All right, good afternoon on this wonderful day. So this is the second last lecture of 2000 and in this cycle of 2012. Next Monday, Clay is going to be talking about learning. And then next year he and I will each be giving our last lecture in this particular cycle, and then we will think what to do for the next year. So today I'll talk about information processing in the brain. I'll tell you about something that some of you might not be familiar with relating to information processing at the level of individual nerve cells in a locust. And then I'll be talking to you about ideas about visual information processing, very specifically in the visual brain of primates, or mammals in general, but more particular primates, monkeys, and humans. So if you cast your mind back to the first lectures we had a fundamental lecture on computation where we discussed what's meant by computation. And although we all have an intuitive sense of computation because we've done it in grade school and we use our computers, it's actually not that easy to define. But in general, it's a mapping from a set of mathematical operations onto a particular hardware that transforms things like electrical charges, like charges of electrons on gates, or ions that have a differential concentration across the membrane in the case of brains, onto new physical states. And the nice thing is you can map this physical representation onto abstract mathematical computation. In that sense, you can say a system computes. And we talked about the idea of universal computation, in the sense of a Turing machine, a very powerful idea that's led in one way. It was all the conceptual undergirding to the information processing revolution and the widespread use of computers. And we discussed the question, to what extent does the brain compute? Because you can, of course, also ask the question, to what extent does your kidney compute? Because in the kidney or in the liver and any other organs, there's also physical operations going on. So in principle, you could also argue, well, that's a form of computation. And sort of I summarize, and I'll try to imbue this with life today, in this bon mot by Shimon Edelman, who's a computational neuroscientist, who said that Alan Turing's legacy and the entire idea of computation can be summarized by observing that just as nothing in biology makes sense except in the light of evolution, so this is a famous quote from an evolutionary theoretician, Dobzhansky, nothing in the cognitive and brain sciences makes sense except in the light of computation. And I'll try to show that to you today. So one very influential way of thinking about computation in the brain is due to two theoreticians, David Marr, who was British originally, but who did his most important work while at the Artificial Intelligence Lab at MIT, together with another theoretician, Tommaso Poggio, an Italian who happens to be my doctor father, my PhD advisor, at the time also at MIT. And they distinguished three levels of analysis, and it's known as a three-level of analysis ideas. One is at the abstract level, there's a computation. If you look at any particular system, be it a computer, or robot, or fly, or human, you can ask, what is the goal of the computation that the system is trying to perform? So for instance, in the case of stereo, you have humans with two eyes, and they overlap slightly, and this allows us to compute depth, so-called binocular stereo. And in principle, and people have built robots that have two eyes, or even more eyes, 3 or 4 cameras, and they try to infer that from the differential position of the image on the camera, they try to infer depth. So in each case, you can say, well, at an abstract level, they're trying to perform this, a similar computation. Why is it necessary? Why do you need, for example, depth information? And what is the logic of the strategy to extract that information? So that's at the abstract mathematical level. Then you can think, well, there's the algorithm. Actually, how do I actually compute this? So let's say I want to compare, the differential images on 2 retinas or 2 cameras. How do I actually do this? What's the best algorithm for doing that? And what is the relevant input and output representation? Should it be bits? Should it be pixels, for instance? Or should it already be edges? Or should it already be corners or some high-level representation? And how do I actually implement it? Do I implement it using what sort of algorithm? And then finally, one has to think about the underlying hardware. So for instance, in the case of a computer, it's going to be digital, it's going to be bits and digital representation or binary representation of information on binary gates. In the case of the brain, of course, it's a very different hardware. And the choice of hardware will, of course, influence what algorithms you use, because certain types of hardware lends itself very easily to certain types of computation, and other ones don't. And even the representations you use is very important. For instance, if you try to multiply numbers in longhand, you can do that using our system of Arabic numerals, where it's relatively easy, or you can try to use Roman numerals, as the Romans did, and it's just not very convenient to do. Of course you can, but it's much more awkward to do multiplication using Roman characters. And so that tells you that the representation of numbers really plays an important role. And so there are these three different levels. When one talks about computation in one mind, you often have to shift between the level of computation, algorithms, and hardware, and they all depend on each other. Originally, particularly David Ma argued that these are independent, but in fact these three different levels are closely related. And here, in fact, these discussion, these metaphysical discussion and computation discussion arose out of many conversation over the course of 2 years between those three. That's Francis Crick on the right, and the middle person is David Ma, who died in 1980. He's written an enormously influential book called Vision, which advocates for the computation approach to vision. And to the left is Tommy Poggio. All right, so as an example, so for instance, to compute motion, we know one way to compute motion. There are different ways. One way to compute motion is to take an image and to take a slightly shifted portion of the image that's shifted that's delayed by a little bit at a neighboring pixel, and multiply those two things together. And you can argue, I'm not going to do that here, why computationally that should be an efficient strategy to extract a motion, to extract in which direction things are moving. Now, one way to do this particular computation, it's one of many possible ways, is, for example, so you have to multiply the image at time x and t with a shifted image at a neighboring location delta x, this is in one dimension, and shifted in time. Now you can do it in this strange way where you exponentiate this expression and then take the log of this quantity plus a log of that quantity. And of course, mathematically they're equivalent, because this property of log, that log of a product is the sum of the logs, right? So log of a times b is log of a plus log of b. Why would you want to do this? Well, it turns out this is an old electronics tricks, and in the 50s and 60s, before digital computer became widespread available, people built analog computers, and due to the exponential properties of transistors or diodes, it turns out this is a particularly easy way to implement this operation in particular type of electronic hardware. Not for us today, because we have digital computers everywhere, but at the time it was. Now it also turns out, and I'll show it to you now, that this is also the way biology does it. This is one way how biology seems to implement products, using this exponential and then logarithmic trick. And so here you have different, so you'll probably, you could use this, but it's not very convenient. You can do it much more directly by shifting bits in this binary representation. You can do it by longhand. You can do it in analog, very large-scale integrated circuit this way, or you can do it in neurons. All right, so we're going to think about the hardware, particular hardware now, and the computation implemented by this hardware. We're going to go to a model system that many of you are probably not as familiar with as a mouse, namely the insects and other creatures, in particular this guy, who's a locust. They're they're very widespread, and their visual system has been studied quite a bit. They exist very often in these very large swarms. This is a plague in Egypt in 2004. Remember, one of the biblical plagues in the Old Testament is, of course, the plague of locusts, and they do actually exist. I've been in one. It's rather scary when you're sitting inside the car and suddenly your entire car is surrounded and the sky becomes dark because there are, a million of these locusts swarming at you. And so what they need to do, they navigate using optical flow, using visual information on their eyes. They have compound eyes, just like flies have. And they're very good at exploiting the optical flow for avoiding hitting each other and hitting their targets. You can see, particularly when it gets very dense, this computation they do very, very quickly, within under 100 milliseconds, up to, with speeds reaction times as fast as 30 milliseconds, really very, very fast. Here's a recording by Fabrizio Gabbiani. You can see. So let's do that again. That's what we're going to talk about. So this is a looming motion stimulus. You can see it started very small, and then suddenly, and of course that's, if you walk towards a wall or something approaches you, that's exactly the stimulus you'll see. And Fabrizio Gabbiani used to be in my lab, in the lab of Gilles Laurent at Caltech. He subsequently is a professor for many years at Baylor, together with Reed Harrison, who some of you will know because he built these beautiful Intan chips that we're also using. They built this little backpack, and so you can record both from the muscle as well as from one neuron. I'll now talk to you about. You have these very powerful jump muscles. So we're talking here about one particular type of neuron called the LGMD, the lobular plate giant movement detector. You have two of them. The animal has exactly two of them, one on the left and one on the right, one each. So two neurons. And it has this very stereotyped geometry. And where you have three dendritic trees, you have this one dendritic tree that's excitatory. You have on the order 15,000 excitatory, in this case cholinergic synaptic input, that impinge onto the dendritic tree. And they map in a very systematic, in a topographic way, just like in a mammal, when you go from the retina to cortex, they map in a very topographic way. They map the visual environment. You have these compound ions, they all map onto this one neuron here. So this is in the lobula, which is three stages into the visual system. There's the retina, the medulla, and then the lobula. And there'll be another neuron on the other side. And then there are three, you can see them as well, there's one here and one here, two inhibitory parts of the dendritic tree that receive 500 inhibitory synaptic inputs. Here you can see the cell body. And this is quite common in invertebrates, that the cell body really is a side, and the dendrite directly terminates into this axon. This is the axon. So the cell body is quite a ways away from, and electrically speaking, in terms of integration of information, doesn't seem to play any significant role. So the spike is initiated here and then propagates down to another neuron. Okay, so it's a very rapid, it's purely feedforward. Today I'm just going to talk about feedforward information processing, both in this system as well as in the brain, in the mammalian brain, in cortex. It's 3 synapses away from the input and 2 synapses away from the output. And turns out this neuron, the LGMD, makes one synapse onto another neuron, single neuron, called DCMD, descending contralateral movement detector. And this one goes directly onto the cervical ganglion, onto motor neurons that initiate, among others, this very powerful jump for the animal to jump away. So since it's more difficult to record from this one, this is a 2-photon calcium imaging that came out in this Nature paper. Typically, most of the recordings are done from the postsynaptic neuron. It's a very powerful synapse that connects this neuron, the LGMD, with the DCMD. In fact, here's a circuit. So typically, people record from this neuron here because there's 1-to-1. Every time you get a spike here, you get exactly one spike here. All right, so this is the geometry. This is the excitatory input. These are two different. These are sort of inhibitory input, and you get integration here, and then you get spikes generation that go down here. Now, if you look at, so in terms of computation, we want to understand what goes on. Well, so here, if you have a stimulus that approaches, so you have a frontoparallel stimulus that approaches you with a particular velocity v, and then with a particular, and so it makes a particular angle theta, and then you can compute, of course, the spatial derivative of that angle, the angular velocity theta dot. So here, what you can see is time to collision, and here's this, the angle that this object subtends on the retina, on the spherical retina, and here is the velocity theta dot in terms of how many degrees per second. And it's a very nonlinear relationship. So as you saw in the movie, first it changes very slowly, but then as it looms very close, it changes very rapidly. Turns out many animals use this so-called time to contact information. When is this thing going to collide with me? People have done this experiments, for example, humans, when they, in car simulators, this is one cue we use to initiate braking, how quickly it looms. There's some beautiful experiments done in gannets, in flying gannets that hover above the ocean, the ocean water, and then they dive down because they see a fish. And you can see the moment they retract their wings is always, independent of the altitude, is always a fixed amount of time before they contact the water. And there's also quite a bit of work done in pigeons. They also use this information to control, and in flies. So it's a fairly common, obviously biological, very important computation. You really need to know this. So the important point is here, it goes very nonlinear. And both the angle increases dramatically as well as the speed increases dramatically. So it turns out so this is the firing rate as recorded from the LGMD or DCMD, which, as I said, is the same. So typically, so these are the different, so then if you look at the geometry of this computation, you don't actually need to know anything about the absolute geometry of the stimulus. It's really all in the relatively relationship between the height to the velocity of the object. So you can all express it as a function of time to collision without knowing the absolute depth. Really, what only matters is how fast does this object, the size of the object relative to the angular velocity. And you can see typically the firing rates are like this. So these are three different values of this relation of the size to the velocity, essentially by changing the velocity. And so typically what you get, the firing rate will increase. So this is time to contact, right? This is 0 here. This is when the object normally would hit the insect. Of course, there is no object to squash the experimental animal. It's all done on a computer, and then that shifts forward in time. And here you can see actually how the angle changes as a function of time. And the important thing here to note that what the cell seems to do, so that's the peak firing rate, there seems to be what happens, the cell is sensitive to angular threshold. So when the threshold subtends a particular fixed value, this value here, the cell, that's always a fixed time before the peak firing rate. So it seems to be that the computation this particular neuron does, or the entire animal does, if the angle becomes too large, then a short fraction later, 50 or 30 milliseconds later, the firing rate peaks just before and then decays. So the critical threshold seems to be the size of the object on the retina. So it turns out you can actually map. So I told you about this trick before because this cell seems to exploit this particular trick. So you can model the firing rate very well by this expression. The firing rate of this cell, of the LGMD, is a product of a term that depends on the angular velocity. It goes linear in the angular velocity. So the faster the angular velocity, the higher the firing rate, times an exponential term negative of the size of the object. So the larger the object will be, the closer it is, the larger this term, it's negative, so the smaller this term is. So that's why you can see there are two contrarian tendencies, right? So firstly, the velocity is small, so it means this term is small, but it also means this one term is small, so this is relatively large. And then as the size becomes larger and larger, this term is going to dominate over this. And so ultimately there's going to be a peak, and then it'll go down to, as this becomes very large, this entire product will become very small, even though this term might be large. Now the question is, how does the neuron implement this particular operation? Well, so the system uses this term, and the long and short of it, this is implemented by presynaptic circuitry. Essentially, this is saturating nonlinearity in the dendrite. This term is simply proportional to how many photoreceptors are essentially activated. The larger the stimulus, the more of these photoreceptors it activates to a first extent. And then this is an inhibition, that's a term. And this term here is, this log is implemented in the dendrite, and then this exponentiation takes place at the spike initiation zone. This turns out postsynaptically to be essentially current. This is a positive current. This is a negative current. The currents are added at the spike initiation, and then there's a nonlinear exponential-like relationship between the kernel and the average firing rate. So that's how this particular cell does it, and it's done at the single cell level. You don't need a network for that. Okay, this is, I mean, very similar to what I showed you before. Here you can see the actual firing rate, and here is the firing rate plus minus standard deviation, and then the red curve is the actual fit. So the expression fits the curves extremely well. So it's a very good model. And here you can see the sort of exponential-like relationship between the average membrane potential and the average firing rate. So this is the actual relationship at the spike initiation zone, and then you can fit it. It's not really, it doesn't fit so well in exponential. It fits a third power relationship between this and this. In other words, the firing rate is the third power of the average membrane potential at the spike initiation zone. So it's very often in biology, these are approximations, and they're only valid over a particular range of parameters, but the approximation is good enough for the animal to survive. And then the nice thing is you can look at all the individual components of this computation and always ask with respect all the individual components of the biophysical circuits, and you can ask what is the function of this and what's the function of that with respect to this particular computation. So for instance, there's a very strong adapting current, a calcium-dependent potassium current. Right? So for every spike that arrives, you get inrush of calcium, and the calcium rushes inside the spike initiation zone and activates a potassium current that counterbalances the excitatory current through sodium and through calcium. And you can see this here. So here, this typically, if you inject current, you get this adaptation, and if you add BAPTA to block this particular membrane, this is good old pharmacology to block this spike potential. Then you get then you remove the adaptation. What's the function of this in terms of the computation performed? Well, here you can see it with and without adaptation. Here, so it turns out that what this this is the same computation. This in this case you always have this looming stimulus. This is the size. They just block it off because it increases so far you wouldn't see the proper scale. And you can essentially see that this computation, if you remove the adapting current by adding this pharmacological blocker, BAPTA, you can see this computation, the peak firing rate isn't dramatically affected. But what this current really does, if you just have translation, because you don't want to initiate constant jumping, right? It's a very expensive operation, right, to jump away. And so you only really want to do that when there's a nice looming stimulus. So it turns out if you just have, if the animal is flying, which of course it does a lot, or you do moving, you're just translating the optical flow, this cell will adapt very, very rapidly. But it will not adapt to these very rapidly changing looming stimulus. So that's the function of this particular circuit for this computation. And so it's very elegant. You can say, well, there's this particular current, the KCA, and its function in this particular computation is precisely to enable it to respond only for looming stimulus, but not for general whole field optical flow type of stimuli. And here you can see experimentally for 3 different values of the size over velocity. You can see the saturation here on a log scale, here on a linear scale. So you have this, remember, you have the excitatory synaptic input in the dendritic tree, and of course you're going to get saturation there, just because these are conductances. And as the conductance becomes larger and larger, it begins to approach the local reversal potential of the associated battery. It's ACH, and so it's going to saturate, and that's exactly what you see. And so this is this log theta dot term, right? So as I said, that these 2 terms, you have log theta dot in the dendrites, and then on the inhibitory dendritic tree, you have a term that's just proportional to the size of the stimulus. But since inhibitory, it's minus, and then those 2 currents, the excitatory current and the inhibitory current, more or less add. You can show that experimentally and modeling-wise. And they go to the soma, and the soma, they exponentiate it again. And so you have this nice computation. It's done at a single cell level. So you got, so this is the operation done on the sorry, this should be theta dot. On the velocity, it's represented presynaptically and then goes through this logarithmic compression stage in the dendritic tree of this neuron. Here you have the term that's proportional to the size, or rather the square of the size. It's just an offset. And then you have the and then you have the inhibition. So then you add up those terms, and then here you go through the sort of potentiation, and that's finally the firing rate you get. So it's a feed-forward system. There's no, well, that's not to say there isn't feedback, but in this very rapid, you don't really have time for a lot of feedback. So it's primarily feed-forward, and does a computation, and does it at the level of a single cell. That's pretty cool. So now we're switching. We're going to continue to look at feed-forward computations, but now we are moving from the Locust visual system to the mammalian system, in particular to the mammalian visual system of monkeys and humans. At this point, although it's clear that, that rats are capable of doing object recognition, there's a beautiful experiment by David Cox and collaborators showing that really very little is known about object recognition in rodents, and I'm not sure anything is known about object recognition in mice. So this is something that's primarily relevant for primates with a phobia. So we are very, very good at doing object recognition. It's our preferred modality, and 25 or 30% of our cortex proper are given over to visual processing in some form or another. And we can recognize, so the average person can recognize anywhere, depending on exactly the age and all of that, 10 to 30,000 visual object categories. Here's a list of one. This was an estimate done by Irv Biederman a while ago. And we're also very good at learning new object categories based on single instances. Yes, Mike?
How do they make such an estimate? What's the essence of how they go about that?
They went into visual dictionary, and they sort of, this was done 20 years ago, and they queried people. So it's not exhaustive, but it's on the order. It's somewhere between 10 to the, it's around 10 to the 4. And a very educated person, or an art historian might know more, a photographer might know more. We're very good at learning new instances from in the limit one picture. So for example, if I show you this picture of this weird animal or whatever on the left, and then I give you these choices on the right, most people will pick one of those two. But based on a single instance. Today I'll show you at the end some state-of-the-art machine vision algorithm by so-called deep learning networks by Google and others, and they typically need hundreds, if not thousands, of example of images. But we, at least once we've learned, right? So of course we, by the time we're sitting here, we've seen literally thousands of hours of visual experience, and we don't come to the world in this way. But once we're over the age of 5 or 6, sort of even children have this ability to generalize from single examples. Look, for example, at Hieronymus Bosch. Look at some of his paintings. You see all sorts of odd, weird animals and demons, and you see one of them very quickly. You can generalize to others. Yes.
That similarity also depends on the context, right? So if you actually change the pool of the candidate, then people may give different answers.
It totally depends on the context, but even without context, where it's the most difficult. Here you're not given any context. I'm just showing you one image on a white background. People are very, very good at generalizing from that. So this is a very famous picture on the left. This is due to David van Essen, who's well known to us, and Dan Fellerman. This is the visual system of the macaque monkey. It's believed that a similar map is also true for humans, but we don't have the level of detail. What it shows you, each one of these boxes, starting in the retina, LGN, primary visual cortex, secondary visual cortex, going up through all these areas and ending on top then in hippocampus, represents an area, a visual area in the brain that's dedicated to some sort of visual processing. It might also do other things, but it certainly involves visual processing. And some of these areas are small, like some of these areas here, frontal eye field. Some of them are fairly large, like primary visual cortex. And this shows you some of the interconnection. Of course, it's quite dated by now, but it's very famous. And so the idea is this hierarchy. You have this hierarchy of stages, and this has been very influential in terms of theoreticians thinking about vision, because a priori it doesn't have to be hierarchical. It could be a random network, Or it could be a very flat hierarchy where you only have one or two layers, like a perceptron, where essentially you have an input layer and an output layer, or maybe three. You have one input layer, one intermediate layer, and one output layer. And mathematicians have shown that under some condition this is universal. Anything that can be computed with this three layer. But here it turns out, there are 10 or 12 or 14 layers. And so people have made, in the computer vision community as well as in the computation neuroscience community, have made models of this. And the general assumption of these models, I'll tell you about their feedforward model. So they have a single pass where essentially you acquire image features and you process it through one or multiple stages. And when you're having many stages, as you use today in today's advanced architectures, you talk about deep learning networks. There's a New York Times article a couple of weeks ago on this. And so the idea is that in this, you have a single sweep of information flow where you here in early stages, you collect a bunch of features like edges and corners and other things. Then you assemble them into some sort of relationships that will tell you there's a person present, or there's Jennifer Aniston present, or there's a bug present, or something like that. But it's all done in the feedforward. Now, of course, we know there's massive amount of feedback. We know that through our own atlases work and through 100 years of neuroanatomy. But for now, these models neglect these attentional feedback or other sort of feedback, except during learning. So typically these networks, I'll show you, they have two stages. The first stage is where the system is learned. That typically takes a lot of time, a lot of computational cycles, and that will surely involve feedback. And then there's a stage once the system has learned, like in our case, we've learned, we've had most of the visual experience, and then the system is very fast, as well as these computers are very fast to operate in a purely feedforward way once they already learned. And so theoreticians separate those two stages. There's no doubt, and everybody admits, that those models ultimately will have to include feedback in real time, like attentional feedback for more difficult to recognize object, right? When it's dark or you have trouble seeing what it is, and you have to look for a while before you recognize it. There's no question that you have to incorporate top-down feedback, but right now the field isn't that advanced yet. So typically they start off with these well-known from the tubal and Wiesel. They start out with two sorts of stages, and so they generalize. These are generalized tubal and Wiesel network. It's a very important insight that people have. And even in the latest and hottest computer vision work right now in these deep networks, or what Google has done, I'll show you at the end, they still use essentially these two stages. They use one stage where you represent particular feature. In this case, early on in V1, you represent edges, right? These are simple cells that Clay talked about in his lectures. And then over here are complex cells. So there's typically a second stage of cells at each layer where you pool over a bunch of simple cells to get invariances. Because here you have a cell, for example, this one, that depends, where the optimal stimulus has to be here or there, right, depending on the phase. Now, of course, you want to recognize something invariant where the exact position is. Well, then you sum over, you do a so-called pooling operation. It's a nonlinear pooling operation, and this is exactly what's being performed by the complex cell. In principle, you can cascade this. You can think of the same two stages of processing in secondary or tertiary or quaternary visual cortex, where you say, well, first you have a stage where input is represented, for example, edge information is represented, and then you want to pool over position and maybe orientation or maybe size, right? Because you want to be selective when there's a small edge or small bar, and you also want to pick up when there's a very big bar. And so when you do that, you have individual simple cells that respond either to big bar or to small bar, but then you want to summate over all of them that have that have the same orientation but different width. Or you want to summate over things that have different colors, or you want to summate over things that have different depths. And so in general, these networks have at each stage, at each hierarchy, they have two stages. They have a simple cell-like stage where they enhance the representation, adding color and depth and whatnot. And then they have a second stage where they summate all that over other variables to get invariance. So here, for example, we know this is a monkey visual system due to John Mansell. So people discovered since 120 years, first based on clinical evidence in patients. Depending where you have lesions, you have different sorts of visual inabilities. So if you have lesion in the infratemporal cortex or the fusiform gyrus or the equivalent in us down here, then what you'll get an inability to recognize things. For example, you get face blindness. You might see there's an eye and an ear, but you're unable to recognize the face, or you're unable to recognize some other visual object. If you have a lesion over here in the parietal system, this system here, you can perfectly well recognize things, but you have difficulty locating them. So for example, you might have difficulty grasping, when you grasp for your cup of water, you consistently miscompute. So this is the visual motor system. So this is also called the ventral pathway because it's on the ventral part, and this is the dorsal pathway. Other names for it are the Watt pathway because this information originating primary visual cortex going down here conveys the information, what object is it. And this is the where pathway. It tells the visual motor system where things so I can reach out and interact with them. And these models here, we're talking about the what system. So it doesn't care, because that's one of the functions of our visual system. I want to recognize Clay no matter where he is in the visual field, whether he's far away or close by, whether he's on the right or on the left side. And that's a very different visual system from the visual system I need to reach out and shake his hand. That system needs to know exactly where his hand is. To recognize his face, I want to be invariant no matter where his face is. And if you go here, if you start off in V1, where the two paths diverge, then you go to V2, V4, infratemporal cortex in the monkey, then you get these more and more complex property. You get receptive fields become larger and larger, so the cells become less, they care less about where stimulus are. So in the limit, you get a Jennifer Aniston cell that no matter where it is in the visual field, the cell responds the same. Well, early on V1, you need to put the bar exactly in the receptor field, and you also get the responses to more complex stimuli. And of course, we know through work of Quinchen Wang and Andreas Burkhalter that something similar, these ventral dorsal pathway also exist in mouse, although they're much less explored. So what people have done, this is, so as I said, this is the work of Tommy Poggio, my advisor, and who's done this over the last 15 years with a range of people, Max Riesenhuber most recently, and Thomas Seri now at Brown. And this model is known as HMax because it, well, for various reasons, it's known as the HMax model. And so here what you see, and here they match sort of, they think these are the areas that correspond to the areas over there. So what's very typical, and like say all these deep learning networks have a very similar structure. So you have a first stage, a second stage, then a third stage or fourth stage, and depending on how, fifth stage. So you start off with a simple cell stage in S1, and then you have a complex cell stage in V1, and then you repeat this operation, but with more complicated configuration of features. But at each of these networks, you have a stage where cells are more selective, and then you have a nonlinear pooling stage where the cells essentially compute something like a maximum operation. So you have a bunch of inputs, and the biophysics implements an operation where only the maximum input is sort of is extracted and sent on in terms of firing rate to the next stage. That's why they're known as HMax. They're conceptually very similar to the classical Hubel and Wiesel hierarchy. And there's a deep literature going back 40 years also within computer vision. There's, for example, the work of Fukushima in Japan on the neocognitron. Yves Lequesne, Jeff Hinton, there's a whole lot of people who worked what turns out in hindsight to be all very similar structures of these sorts of feed-forward networks. So there's no feedback, and you have localized receptive field. In other words, this cell only gets input from a local part of its visual, it doesn't get input from all the previous layers, but only this. And this cell will only get input from a very limited part of the previous stage, not from all the network. So it makes it local, makes it easier and less expensive to compute. It minimizes wiring, and of course that's what we observe in the visual system. Yeah, so it's there's lots and lots of literature in this. There's sometimes also some versions are called convolution networks or deep learning networks. So it's a different view here of the same pyramid, where here you have simple edges. Here you pool over the edges complex cells. Here you do configuration of simple cells. So in the second stage, use configuration of edges like this or like this. You get corners or Mercedes-Benz triangles or other things, until finally you reach a stage where you represent things that we are cognitively familiar with, because we don't see the world in terms of edges and corners. We see it in terms of people and dogs and bananas and stuff like that. And so here at the last stage, you have a so-called network that expresses particular categories of objects, like buildings, dogs, people, faces, cars, etc. And then, of course, then the claim is, so these networks only model this stage. What you then want to do, but the field hasn't advanced that much, computer vision. Ultimately, of course, you want to now, so you know there's a banana out there and you know there's a dog out there. Then, of course, you want to do what some people like Shimon Ullman call visual routines, because now you want to do something with that information. Where's the animal? Is the animal threatening? Is it my pet? So you want to do all sorts of operations on this. And of course, we know from monkey work, in particularly the anterior form of IT, there you have these high-level cells. And of course, in humans, you have even these even more high-level, these Jennifer Aniston cells. So the architecture is as old as Hubel and Wiesel, 50 years. As I said, you have simple cells that are selective for orientation, spatial tuning, wavelengths, etc., depth, if you want to include stereo. Then you have a complex cells where you pool over the location, orientation, spatial tuning to get invariance. So each simple cell stage increases the complexity of the representation, and each complex cell stage implements tolerance to one or more features and thus leads to generalization. Yeah, so there's a whole bunch of these models. So here, for example, at a higher order stage, first you have these simple cells in V1, and at a higher stage you have simple cells, let's see, in the model in V2 that represent, this inverted T at different locations. And then you want to have at that stage, you want to have a complex cell that, for example, represents the existence of a T independent where it is in the receptive field. So here you have a complex cell that sums over both location as well as size. So it's tolerant. So this cell now is tolerant to the exact size of the object and to its exact location. Of course, that's what we see in cortex. So there have been done experiments in macaque monkey by Locust, where the monkey grew up with paper clips, with particular forms of paper clips, but only one particular size. And then you can, even though the monkey is only familiar with one size, with images of one side of paper clips, you can show that once the monkey has learned that, the cells are invariant. Seems to be a property that the monkey has learned very, very early on. And so now these cells in IT they don't tend to care about size anymore. So you need some sort of operation like this. So early on, the typically Gabor function. So this is a type of function that was first described. Clay talked about it in cats and then in monkeys. So here you have a receptive field of a simple cell in a primary visual cortex. Cat or monkey, it's very similar. And then you can model them by these products of essentially Gaussians with either cosine or sine term depending on the phase. And so you can do that all over the visual field. You can assemble them into these so-called ice cube models of Hubel and Wiesel, where you pull them from the left eye and the right eye, and you do it for all different orientations. So finally, in these implementation, you get models like this in early stages, where here, for example, you have a simple cell that is this particular horizontally, but exists at many, many different sizes. And here you have another simple cell that is, diagonal, but again at many different sizes, because the world, things are different sizes because I don't know how far they could be, right? So, if this object is twice as far away, of course its size is reduced by a factor of 2. So you need to have a mechanism that's tolerant to size. And then you also want the mechanism that's tolerant to the exact location. So that's why you need this pooling stage. All right, so a little bit more in detail. So in each stage, whether it's early V1 or at higher stages, you have this sort of rectification and squaring. So you take, this is just an iconic representation similar to the one Clay showed you of the Gabor function. You square the output essentially of different phases to be invariant, and then you do what's called a divisive normalization, so you're invariant to the contrast. The visual objects can vary enormously, and even the same object, if you take this object, depending on where it is and whether it's bright light or moonlight or sunshine light, it can vary enormously, so you need to normalize for all of that. It seems trivial, but this actually has plagued computer vision for decades, because objects, even though to you they appear always the same, if you look at the level of a camera or level of photoreceptor, depending where it is in the light, etc., they can look very, very different. So you have to compensate for that. And there are these stages. Once again, so for example, you want a stage early on that's where the cell fires to this bar and the A independent where it is, and you want it to fire for small a or for big A. And then finally you get responses like this. So this took a long time in computer vision. It's called a bag of features. So for many, many years, and some computer vision still is like that, the way people recognize a giraffe or face is to look for exact spatial relationships. So you want one eye here, so you want a mask where you have two eyes and a nose and a mouth, where the geometric relationship has to be exactly preserved. And some models do that. They tend to be very expensive because you need to do this operation everywhere in the visual field at all possible scales. The other set of models that are like this here, that are probably, they're much more biologically like, don't exactly care where the exact, so even if you had a Picasso face, where everything is slightly changed, these cells would still respond because they don't care about the exact spatial configuration. Because doing so is very expensive. And so these are more bags of features. As long as you have eyes in some neighborhood, you have two eyes and you have a nose and a mouth, the cell will recognize it as a face, rather having to be very demanding in the exact location of the. So that's why you get the cell that responds, these cells that respond to giraffe. And now you have a simple category that says, well, in the presence of eyes and this funny shaped neck, this typical leopard spot-like net, then you're going to have a cell that tells you there's probably a giraffe out there without having to fulfill exact spatial relationships. So one of the early tasks these models were applied to are these tasks by Simon Thorpe, who lives in Toulouse. He did the following. People are very good at this. You should all have no trouble in recognizing that half of these images roughly contain animals and half do not, right? Now I can do it much faster than here through. You can do this essentially 10 milliseconds. And these are images you will have never seen. And you can take totally untrained people off the street. And we all are very, very good at this. I mean, this is what our visual system grew up with. And I can also ask you, is it a truck or not truck? Is it male or is it female? You're very good at doing these sorts of tasks. Color, even if they're black and white, performance is the same. So this was a Nature paper, came out in a Nature paper, because what they did, it's a very simple experiment. They then looked at the EEG of these subjects, and they compared the EEG when you, for example, when you look at all the images when there was an animal, and then you subtracted the EEG, the evoked potential, from all the images when there was no animal. So people are essentially 100% correct. And so then you subtract those two EEGs, you get the average response to an animal image and the average response to non-animal image, and you subtract the two. And what you see, depending on which electrode you do it, what you see is starting 150 milliseconds after the image onset. So the image comes on here, then 150 milliseconds after that, the traces begin to diverge. So what that tells you, that somewhere in the brain, something in the brain knows roughly, or starts to know after 150 milliseconds whether there's an animal or not animal present. And as I said, if you do the same thing, if you ask, do these images contain a mode of transportation, like a truck or a canoe or an airplane, versus no mode of transportation, it's exactly the same. So between 150 and 180 milliseconds, your visual brain knows already that there's an animal out there. Now this is puzzling because it's very, very fast. So this is a rendition of the visual system. This is now applied to human brain with some rough response times. So you go here, you go through the LGN depending on the brightness, of course. But at these brightness, between 40 and 50 milliseconds, information will hit the LGN. Then a mere 10 milliseconds later, it's in primary visual cortex, 50, 60 milliseconds. And it gets to IT maybe at 100 milliseconds, 120 milliseconds. Later stages maybe 140. Hippocampus typically 300, amygdala 350 milliseconds. So those times are slow. Well, remember, those times is the onset of the response, those times here. So in other words, it takes your brain we know this by EEG, no matter how the brain does it, between 150 and 180 milliseconds, something in your brain knows already the answer. And so given these delay times, it doesn't leave you time for a lot of iterations. So this was a very powerful argument for why at least these tasks, like is an animal not present, is done in the first wave of spikes. You have this first wave of spikes. Francis Crick and I call it the net wave. The net wave that comes up in the retina, sweeps through the LGN, goes into V1 and down the ventral pathway. In that first wave, essentially, you make the discrimination because there isn't time for a lot of iterations. Right? And these models fit that data very well. So this, for example, is this H-max model, which was specifically applied to that data, where you have a small number of categories. So you have animals versus non-animals. And you look at this is D prime, so this is a measure of performance of human observers versus a model observer. And depending what you have, whether you just have a headshot, or whether you have a close body or far body, where your performance, I mean, here, the lower the D prime, the worse the performance. And the model, both the model and humans behave very similarly. And the interesting lessons here that these models also do quite well. In fact, that's basically state of the art to engineering models that don't care at all. You know, when you take a hard-nosed engineer, it doesn't care at all about what the brain does. They just want to do the best possible job. And if you compare them head to head, they essentially do. So this is a model applied. It's funded by DARPA, applied to, in effect, now fielded out there. These models, you know, they do all sorts of things that DARPA is interested, Defense Department is interested in. In fact, I wanted to give you a state-of-the-art machine. So this just came out a couple of months ago, hit all the newspaper because it involved Google. In fact, the networks are really very standard I mean, there are a few groups that have done this, not in this spectacular fashion, but the idea themselves is as I mentioned, always the same idea. This is done by Andrew Ni and NG at Stanford, together with people at Google. They took advantage of the Google planetary resources. So what they did essentially, they have a network like the one on you see that in the image, which they have 3 times, they have these layers and then they replicate this 3 times. So that's why it's called deep learning. There's multiple layers. But the architecture is very similar. It's feed-forward architecture, and this stage you know, feeds into the next stage. And you have a simple cell stage and you have a complex cell stage. And you, here the remarkable thing is because they had 16,000 cores that they could use for 3 days non-stop computation, they actually fed it 10 million images they grabbed off the web. These are some of the images. Just you know, they just grabbed them off the web, and normalize them to 200 by 200 pixel images. And these are unlabeled. That's very important. So the computer did not say, or a human didn't say, okay, that's a horse, and that's a newscaster, and that's an urban scene, you know, that's a toy, etc. But so what you're doing, this is as I mentioned before, there are 2 stages for these networks. You have a learning stage and a recognition stage. So here, sort of standard computer vision, or at least in the early days, you had to label images. You had to say, well, this is a face, or you have to really outline the face. With these algorithms, you don't need that anymore. You just show the computer 10 million images, and then it'll figure out by itself, oh, there's something common about a whole set of images that look like this. And they're very different from images that look like this or that look like this. The different configuration of edges. The system figures this out by itself if you show it enough example. And surely that's what our system must do, you know, as young babies. And then you test it with a different set of images. You can't test it on the same ones, again unlabeled, and you see how good this is. So it's very high. If you just test it against faces, it's like 90% correct. What's more interesting, if you test it against a database of 10,000 object categories, where you have pencils versus cars versus trucks versus airplanes versus people's hands, its performance absolutely is still very low. It's 16%. It's pretty low. However, that's 70% better than state of the art. So right now, the state of the art, if you want to recognize general purpose objects like we do all the time, we are still much, much better than any computer system. Computer systems essentially are as good as some people argue better than us for very specific categories. In particular for categories of handwritten numerals and for faces. So for instance, if you go to the border between Hong Kong and China, which is apparently one of the busiest borders on the planet, all the passport control by and large is now computerized, and these systems can store 50 million images of people, and they can match any image against 50 million. Of course, no human can do that. Where we are still much, much better is sort of generalized vision. So this is a state-of-the-art system. It's better than any other computer system, but it's still performance is far away from human. But then you can look at for individual images, and you can look at some of the neurons. And then, for example, they find one neuron that preferentially responds to these things, which turns out to be cat. So in other words, from feeding it 2 million images, 9 million images, the system, one particular neuron, specialized to respond to this. So that's like a cat image. Another neuron only responds to this. This is a human image, right, of a silhouette, because many of the images contain humans, of course, probably more than half. And so a cell learned by the network learned by itself to wire itself up to only recognize, to human, to the human form. And so you can, well imagine how these same sort of algorithms operate in our case. So in summary, feedforward architecture, although we all know that their approximations to the real thing seem to be very powerful and seem to be a powerful way to solve the problem of object recognition. They're compatible with the anatomy and physiology of visual cortex. They have these simple and complex stages, but we all know they're incomplete. One problem is they suffer from so-called clutter. So all those images were small images where typically you only have one object, or you have a whole collection of objects like just cars or just toys or something. But for example, if you had two objects, if you have my face versus a cat in the same image, then performance begins to suffer because my face will act, if you have a cat neuron, that'll tend to reduce response, versus if you have a human neuron, it'll tend to reduce by the cat. And so if you imagine, but we're very good at recognizing things in clutter. And so this is a problem that computer vision really, they're trying to address, but hasn't been solved. These problems, these networks right now cannot learn from similar examples. As I showed you, you have to feed 1 million of images. But of course, everybody neglects the feedback, and so this may be, particularly the clutter problem, that might be addressed by shifting things around, by having a spotlight of attention where you just focus on the cat, or where you just focus on the person. So I hope to give you some feeling of information processing in visual system, which is after all the one we have chosen to look at here over the next 10 years. Thank you very much. Yeah, and people have looked at that. So it really depends on what type, there are all sorts of illusions, right? There's object invariance illusions. So for some illusions, these networks work well, but for other they don't work well. So one really has to go through specific classes of illusions. In principle, I don't see any a priori problem why these networks shouldn't suffer from the same illusions as we do. And people have looked at that, yes. Yeah, so in a sense it's mysterious why, so it's not a priori clear why these systems work so well as they do. It's not really clear. So people have made this discovery, and it's what remains to be discovered. Tommy Poggio says he has a theory, and he talked about it here at the last symposium, to explain why the remarkable good performance of these, after all, very simple networks that have one or two operations replicated, 6, 7, 8 times. We don't, at this point, they seem to work remarkably well. We don't really have a good understanding why. So you need not only models, but you also need a deep theory. And it'll probably come from these learning people, from Jeff Hinton and people like that, or Stephen Smale, the mathematician, I know he's working on it, to explain why do they work so particularly well, and probably has to do with the structure of the natural world. Because you can certainly imagine artificial worlds where this may not, where this is not the case, but the real world, the natural world, which is all sorts of structures after all, and power laws, dependency, that's probably one key explanation why these work so well on natural images.
Do you think this is something that neuroscience could potentially?
Well, yeah, I think this is the same trick that neuroscience exploited. That's why I think that's why you have 12, if you look at if you remember the Van Essen hierarchy, that's why I think why we have 12 stages in our object recognition pathway, because we want to recognize things independent where they are, and I think that's the reason. Well, because you need to have two things present. So one thing, you have local receptive fields. So, if you want to, so let's say extreme case Jennifer Aniston, you cannot have in the retina Jennifer Aniston neuron, because you want to find her independent where she is, and you want to find her independent of the brightness, right? And independent whether you have one eye or two eyes. And we can do object recognition with one or with both eyes. So by the time you have all these invariances, you need all these computation that are invariant to the position, to the brightness, to the scale, far away or close by. And so in principle, mathematically, you could compress that all, but it turns out since you want local neurons to minimize wiring, you need multiple stages. And it's just, for those reasons, I think the brain is implemented in a hierarchy. And insects, they don't have that. They don't have deep learning because they don't have 8 stages, and by and large, they don't have the ability to recognize objects with invariants that we have. Well, so I should take back what I said. I know at least in one case you can train bees to recognize individual faces. People have done this behaviorally. So you put up different faces and you have a little sugar in front of them and they'll only fly to one. So I guess at least presumably in the mushroom bodies they can somehow do that, and I have no idea how, do they use the same trick, and how maybe they can only do it for 2 faces but not for 10,000, I don't know. It's a good point. So I don't know how insects do it. But I've never seen any behavioral evidence that they have the flexibility and capability that we have, that we or other primates have. And it remains to be seen by us how good are mice. Can they recognize different sorts of camembert visually, for instance? One of my favorite experiments I like to do. Or Bordeaux wine. All right. Thank you very much.